Ablation of either p21 or BAX prevents p53-dependent apoptosis induced by green tea polyphenol epigallocatechin 3-gallate. Treatment with epigallocatechin 3-gallate, EGCG, a polyphenolic compound of green tea, results in activation of p53 and induction of apoptosis in prostate cancer LNCA P cells. However, no direct evidence has delineated the role of p53 and p53-dependent pathways in EGCG-mediated apoptosis. To understand the mechanism of negative growth regulation of prostate cancer cells by EGCG, we undertook a genetic approach and generated an isogenic pair of prostate carcinoma cells PC3, p53, by stably introducing a cDNA encoding wild-type p53. Treatment of the resultant cells, PC3-p53, with EGCG led to, as reported earlier in LNCA P cells, an increase in p53 protein, which exacerbated both G1 arrest and apoptosis. This response was accompanied by an increase in the levels of p21 and BAX. The cells lacking p53 continued to cycle and did not undergo apoptosis upon treatment with similar concentrations of EGCG, thus establishing the action of EGCG in a p53-dependent manner. This observation was revalidated in another prostate cancer LNCA P cells harboring wild-type p53. Inactivation of p53 using small interfering RNA, siRNA, rendered these cells resistant to EGCG-mediated apoptosis. Because p53 activation led to increase in p21 and BAX, we investigated whether these two proteins are important in this process. Ablation of p21 protein by siRNA prevented G1 arrest and apoptosis in PC3-p53 cells. The p53-dependent increase in BAX expression altered the BAX-BCL2 ratio and paralleled the activation of caspase 9 and 3 in cleavage of PARP. Transfection of cells with BAX-siRNA abolished these effects and inhibited apoptosis but did not affect the accumulation of the cells in G1. In summary, using isogenic cell lines and siRNA, we have clearly demonstrated that EGCG activates growth arrest and apoptosis primarily via p53-dependent pathway that involves the function of both p21 and BAX such that downregulation of either molecule confers a growth advantage to the cells.